Today we will do the blood smear preparation. My young scientists, the material required for the blood smear preparation consists of any type of dropper or for the correct measurement we will use the micropipette, tips for taking the blood, there is an instrument and the pins, alcohol swab, slides and the cover slips, methanol, 99% methanol, Giemsa stain, water. We will visualize our slide under the microscope. So first we will record our experiment in our logbook, which is an important bioethical measurement for the lab. So first we will write the date, the time interval and the other main important details we will want to record it. And obviously we may have also, it is important that we should have the data book or the practical copy. Now I will start the procedure. First I have to disinfect totally and clear my finger. I will take a clear slide which is also cleared by the ethanol and will put a drop of blood. I cleared the finger. Then I will take another clear slide and at the angle of 45 I will make a comet-like shape. First I have to fix, then I have to drag it. You can see here a clear comet-like structure has been formed. I have to dry it. So before going on further steps on the procedure, I will wear the gloves. The first washing will be with the methanol. So that we have to fix our slide. In methanol, we have to fix our slide for 2 to 3 minutes. We will give the washing of Giemsa stain. We will wait for about 5 minutes and transfer it to the another clear petri dish. After 5 minutes, I will wash it with the water. So I will put this dry slide under the microscope. First I will adjust it at the lowest magnification power, it is the 4x. And one important measurement which I have to do here, I will see the slide in a zigzag manner, and we want to see each and everything, then we have to go just like this. So we have to cover each and every cell. As the blood cell is consist of different type of, not only the red blood cells but also the white blood cells. In the slide we can see the biconcave shape of the blood cells and various types of the white blood cells, which consist of macrophages and the neutrophils, eosinophils, basophils. Depending upon their shapes, we can see here and classify these cells. Along with these, we can also see here the platelets. On 4x, I can see there are various small spots of the red blood cells. So with the more magnifying power, I can fix it. I am using 10x magnification power, and these cells are more beautifully visualized to me. Then I am changing the 40x, and at the 40x I can see by moving the slide various blood cells, and more perfect view I will change the lens, the objective, the 100x, and for visualization, better visualization, I will use oil immersion. 1 drop of oil immersion is enough. So, my young scientists, the blood smear preparation experiment is very important, not only for the medical point of view, but also in the biology lab. We can see and compare different types of the slides which are of the different species, which may be of different abnormalities. So it is very important experiment. Thank you.